Hello, and thank you for joining today's event with Calidi Biotherapeutics, which trades on the New York Stock Exchange American under the ticker CLDI. With us today, we have Andrew Jackson, Chief Financial Officer, and Guy Travis Clifton, the Chief Medical Officer. We will begin with a brief presentation in a moment, and then we will answer your questions. Users may submit a question at any time by using the Q&A tool at the bottom of the Zoom window. Before we begin, please allow me to read the Safe Harbor Statement. This call may contain forward-looking statements within the meaning of the Private Securities Litigation Reform Act of 1995. All statements pertaining to future financial and/or operating results, along with other statements about the future expectations, beliefs, goals, plans, or prospects expressed by management, constitute forward-looking statements. Any statements that are not historical fact should also be considered forward-looking statements. Of course, forward-looking statements involve risks and uncertainties. I now turn the webinar over to the Calidi team. Gentlemen, please go ahead.
Thank you. Good afternoon, good evening to everyone joining. Thank you for your interest in Calidi. I'm Andrew Jackson, the Chief Financial Officer. I'll be introducing you very shortly to Dr. Travis Clifton, who we're very excited has joined us as Chief Medical Officer. You should see the presentation up on the screen. So we are an immuno-oncology company with a systemic and localized approach to attack cancer. We have our own forward-looking statement and risk factors. So this one slide really encapsulates what makes us different from our competition. On the left, you'll see what we call a naked virus. So this is a virus, an oncolytic virus that's unprotected. And as you'll see, it gets rapidly eliminated by the body's immune system before it reaches the targeted tumor, leaving the tumor largely untouched. On the right, what we do is load our virus into a stem cell, and this does four things. One is a stem cell protects the virus from the body's immune system. Second, it allows the virus to amplify and potentiate inside the stem cell. Third is it actually opens up the tumor microenvironment so that the full payload of the virus can attack the cancer. And fourth, it activates the body's immune system to recognize and target and attack cancer in the future. That's the platform that we built the company on. And what's been really exciting recently is we've been able to take that same concept of protecting the virus and shrink down the form factor. So on the left, what you'll notice is we actually envelop the virus with the human cell membrane. And what this allows us to do is deliver it systemically. So we can deliver it through the bloodstream. And with this indication, we're going after lung cancer and metastatic cancers. We have some value inflection milestones coming up in the rest of this year, starting with CLD 400. So that's RT Nova, that's our systemic platform. Our post has been accepted for AACR, and we'll be giving a platform update there next week. Then also, we'll be selecting our lead candidate, and we'll be displaying that at ASCO in mid-year. And then we also expect to be having a Type D meeting with the FDA for RT Nova around mid-year. CLD 201, that's solid tumors. We submitted our IND in March, and the FDA accepted it in April. So we have an open IND. We expect to dose the first patient in the third quarter of this year. Then CLD 101, that's newly diagnosed high-grade glioma. We have Northwestern University has an open IND, and we expect the first patient to be dosed in the second quarter of this year. And then finally, for recurrent high-grade glioma, we announced that we had enrolled 14 patients at City of Hope earlier this month, and we expect to be doing a biomarker study update a little bit later this year. So to go into more detail on our mechanism of action and to introduce himself and tell you a bit about his background, I'm pleased to introduce Dr. Travis Clifton.
Hello, my name is Travis Clifton. I'm a newly joined as the Chief Medical Officer of Calidi. I'm a practicing surgical oncologist. I have 18 years of experience in both translational clinical trials and drug development. I've previously been the CMO of other companies to include Incendia Therapeutics, which was formerly Parthenon Therapeutics, and I was a co-founder of that company. And then in a consulting role, I've helped many companies throughout the years for drug development. For my background, I did undergraduate education at West Point, then medical school at Vanderbilt, did my surgery training in the military in San Antonio, and then surgical oncology training at MD Anderson. And then I've been a practicing surgical oncologist for the past 9 years. So to talk about Archinova, what's really exciting about this is that it's a systemic virotherapy. So it both allows systemic administration, which opens up the potential treatment for this virotherapy beyond what is intratumoral injection, but also importantly, it is designed to home in and self-amplify in the tumor itself by avoiding the immune system, which is sort of a problem with systemically delivering a naked virus. And then beyond that, we can modify this virus to deliver any number of payloads, which really makes it a very versatile therapy. So the applications to me are quite broad and very easily combined with other therapies as well. Andrew, you can go to the next slide. So to go through the mechanism of action first, again, this is an enveloped virotherapy, so it's able to avoid the rapid clearance from either antibodies or complement that a naked virus would be prone to. Then it's has selective viral amplification and spread within the tumor microenvironment. So again, it's delivered systemically. Anywhere that there's tumor, this virus will localize to and amplify in that environment. That, of course, by being nature of being a virus, it's naturally immunogenic, releases a number of factors that help drive the immune system while also causing oncolysis that releases tumor-associated antigens into the tumor microenvironment. So in that inflammatory milieu, you get recruitment of immune cells that induces anti-tumor immunity, which includes infiltration of T cells into the tumor microenvironment while decreasing the immunosuppressive subpopulations like regulatory T cells or myeloid cells. So you create a robust anti-tumor immunity that leads to tumor killing. So all those are sort of inherent to oncolytic viruses in the tumor. What's really, to me, very exciting about this is it also allows us the delivery of gene payloads, which is very versatile. But these are things that we can selectively deliver to the tumor that could be immunomodulatory or existing anti-cancer therapies as well. Think of things that are potentially too toxic to deliver systemically. Now we can localize them to that tumor microenvironment so they can augment or work in addition to the natural mechanisms of an oncolytic virus to really amplify the therapies, and also can be tailored to different indications. Ultimately, this leads to mass elimination of the tumor and effective therapy. So I'd just like to highlight Supernova. So this is the trial that's beginning. Again, as Andrew mentioned, the IND for this has been accepted. There's previous experience with a predecessor version of this that was both safe and tolerated with strong signals for efficacy. In this example, this was a thyroid cancer that was treated. This is published data with complete resolution of this mass in a short period of time, and that was with a single injection. So we've really taken great efforts and pride in really perfecting this in a way that it's off the shelf, can receive multiple doses. Again, we have confidence from our prior experience that this will be both safe and effective, but this will allow it to reach a broader population. So that phase 1 dose escalation trial, again, is hopefully starting at the first half of this year. That's the plan. Importantly, we have already kind of honed in on which clinical sites we want to use and what our indications, most interested indications are. And so those include head and neck cancer, triple negative breast cancer, and sarcoma. The initial part of our trial will be in all comers of advanced cancers, and so we'll get additional experience with that. We have some thoughts on potential other indications, so um, we're excited to finally dosing patients, and we think that we'll have exciting results from that. So just to go in a little more depth on what the phase 1 trial for this looks like. The first part of the trial is a classic 3+3 design. So that's we enroll 3 patients. As long as there's no dose-limiting toxicities, we escalate to a next higher dose. If there are toxicities, we would enroll an additional 3. We're testing 4 dose levels. Again, in this first part, we'll be enrolling patients of advanced malignancies of all histologies, although we've already working with our PIs to help optimize patient selection even in this first part of the trial. And the objectives of this are to confirm safety and then ultimately determine the optimal dose for the second part. That second part will include, right now, we're planning for 10 patients of 3 or more histologies. As I talked about previously, the areas we're initially focused on are head and neck, squamous cell carcinoma, triple negative breast cancer, as well as sarcomas. And the purpose of this is really to refine the indications. So beyond just the indications from a histologic standpoint, we can really drill down on patient selection by having more experience, as well as to understand the expected response rate, which will help us adapt part 3 to make sure that we have enough experience and really can adequately power part 3, potentially to be a pivotal trial. In some rare indications, even in a relatively small trial, you can go from to BLA from there, but that would depend on which indication we're in and how robust the responses are. There is possibilities to help satisfy Project Optimus and for ourselves that we could test additional doses or even combinations in part 2. And so those are things we're actively looking into now.
Great, thank you, Trevor. That concludes the formal presentation. Happy to open up to questions.
And I'll just remind everyone that if you wish to submit a question, press the Q&A button at the bottom of your Zoom window and type your question into the text box. Dr. Clifton, we had some questions for you here. What excites you most about Calidi's approach to treating cancer with targeted oncolytic virus therapies, and how do you see this transforming the standard of care?
Yeah, it's a good question. What really excites me is that virotherapy has been around. We know that it works. That's been shown both in our own personal experience with it, but there is approved therapies in that space. But they aren't used broadly, and primarily that's because the efficacy is somewhat limited by the rapid clearance of the viruses from the immune system. So it works. However, you don't get long-lasting or potentially deep therapeutic responses because the immune system clears it. And if you inject it the first time, your immune system is naive to that. So it's a setting where it may work initially, but however, as the immune system becomes more primed to that virus, it clears it before it can have its oncolytic effect. So I really think Calidi's approach to protecting and amplifying the virus will help us to really reveal and determine the full potential of virotherapies in a way that I think is potentially transformative. And then for RT-Nova in particular, I think that the modularity of it, so the ability to adapt the payload, really is going to open up a whole new world of potential for virotherapy beyond what even we're currently imagining or have based on prior therapies.
Sticking with RT-Nova, Dr. Clifton, could you elaborate on how RT-Nova overcomes the limitations seen in first-generation oncolytic viruses?
Sure. RT-Nova overcomes those in several ways. Um, One, first and critically, it protects it from the immune system. So it's enveloped in a way that it can both circulating monoclonal antibodies circulating antibodies in general cannot clear it. But additionally, complement cannot clear it as well. So it's able to get to the tumor. If you injected a naked oncolytic virus, it may never ever make it to the tumor microenvironment because of that. The other thing that's important about it is it's systemically delivered. So intratumoral injections certainly have a role to play and are an important therapy, but there are going to be some patients who have either too many lesions or lesions that are not in injectable locations. And so that inherently limits an option that's intratumorally delivered. So having that systemic therapy um, really opens up the potential of oncolytic therapy to a broader range of patients. And the other is, that also fits into a paradigm that's often used for cancer drugs. Obviously, systemically delivered drugs are used for a variety of things, so that I think will help um, open up adoption as well. And then third, I think I touched on this, but the payload for it makes it extremely versatile in a way that it can be tailored to specific tumor types, opens up opportunities for collaborations with others. So I think that will really elevate the options for virotherapy broadly.
Now, from an investor's perspective, what makes Calidi's pipeline and research uniquely positioned for commercial success compared to other biotech firms in the space?
Well, I think, the strength of the underlying science to me is sort of the key. Um, the Calidi has taken a very rigorous scientific approach to incrementally advance virotherapy. We have the right people and have done the right background work to really have a well thought out both pipeline that's complementary, but also kind of we have the advantage of following on and learning from the attempts of others before us, both what's worked and what hasn't worked to adapt our technology and our pipeline to really capitalize on that. So I think it puts us in a key position to really advance virotherapy in a way that again is I think potentially transformative in cancer therapy more broadly. So um, I think it's at the end of the day the rigorous approach we're taking and that we've assembled the right people to really learn from what's come before us and make the most of it.
How do you plan to navigate regulatory approvals and market adoption for Calidi's therapy?
Well, I think from that standpoint, to me, patient selection and indication selection are really critical for this. At the end of the day, regulators want safe therapies, but they also want what's best for patients. And so being able to demonstrate robust and reproducible results in the clinic will really help clear both regulatory hurdles, but also, um, you know, lead to rapid market adoption. Um, I think that we've taken a very rigorous approach to safety, both in our preclinical work and in our clinical trial design. So that will help to address regulatory concerns of giving a viral therapy, even though that's been done many times. We always want to make sure that this is a safe product. Um, and again, the attention to detail in the clinical trial design that makes sure that we can reveal unforeseen toxicities that might, that's always a potential risk, but something that I think we've really taken great efforts to mitigate by appropriate trial design in a way that's adaptable to address those. So again, things like dose selection, choosing the right clinical sites and the right PIs are really critical for this. And these are things that we have not taken lightly, so that ultimately Having these right PIs to talk about this success will help, both make the trial run more efficiently, but also lead to um, rapid clinical adoption as well once the results are in.
Can you share your vision for upcoming clinical trials and key milestones that investors should be watching for?
Yeah, so these trials, inherently take time. They are designed to be safe and incremental. So, the first part of the trial is focused on safety and dose selection. And so, we have designed the trial in such a way that it will both move as quickly and efficiently as possible and be as adaptable. Because undoubtedly, when you're doing these trials, you learn lessons as you go. And so making sure that that trial adapts to those, but does it in an efficient way where we don't have unnecessary pauses, um, will help us to um, to move quickly towards commercialization. But I think you can expect us to talk about the safety and the dose selection when we get those done. And then additionally, ultimately clinical responses and indication selections, which will help the investment community understand where our market is and the size of that market and things like that. So, um, we are always trying to balance both for investors, but also ultimately to get this drug to patients as quickly as possible. What's the fast to market approach? And then also, where's the approach to getting this therapy to as broad a population as possible so people can benefit from it. And sometimes those things aren't the same. So there may be both a fast to market strategy to get the drug approved as quickly as we can, but that may be in an orphan indication where the market is relatively small. Meanwhile, that's coupled with a more broad strategy that may take longer, may require randomized trials and things like that, but opens up the possibility of having this treatment available to a broad number of patients.
The data show robust immune modulation across tumor microenvironments. From a clinical development standpoint, how are you planning to incorporate these biomarker-driven insights into patient selection or stratification in upcoming trials?
Yeah, that's a good question. So I think, understanding the how the drug works, that's determined from these biomarkers and the preclinical work. Um, but part of it is understanding which patients we're enrolling. So we are taking patients who have historically have cold tumors or tumors that are unresponsive to current immuno-oncology options or at least no longer responsive to them. And so there's some of that that goes into which indications and patients we select as from a biomarker standpoint. And then critically, we've developed a biomarker strategy to complement our clinical trial so that we can get as much information as we can from each individual who, graciously enrolls in the trial to make sure that we, not just understand the clinical response, but have the biomarker data that goes with that. So things like making sure that we are modulating the tumor microenvironment in a way that we expect and that we can tie back to our preclinical biomarkers. And undoubtedly, we will learn things from that you know maybe are different than what we expect, but that doesn't mean it's bad. It means that we can help understand optimally which indications and which specific patients are more likely to respond, and have a biomarker driven approach that could mean that we have novel tests for patient selection along the way. But I think most likely this will be, effective broadly aside from that. Um, as part of that, we've really prioritized obtaining tissue biopsies from both injected and non-injected lesions as part of this trial. And I think that will help us to understand and correlate those preclinical results effectively. Obtaining tissue in a phase one clinical trial is always challenging, but I think, one thing that's unique to me about this trial is because these are intratumoral injections, we will be able to get biopsies, needle biopsies, probably at a higher rate than in historical clinical trials for things that are delivered systemically. So I'm excited about that because, ultimately you learn a lot from those, both from drug mechanism, but also patient selection to help adapt the trial as we go.
Calidi is pursuing both systemic and localized approaches. How do you see these strategies complementing each other? And how might this dual pathway approach shape Calidi's positioning in a competitive immuno-oncology landscape?
Yeah, I think these strategies complement each other very well. There's unquestionably a need for more localized therapies. I'm a surgeon. When patients come in with localized tumors, the first thing we typically do is to cut them out. But there are lots of patients who you can't do that to, even though they have cancers that are either don't have the ability to spread or have not gained the ability to spread. Some patients, for either anatomic reasons, it's wrapped around vessels and the things that you can't remove, or patient tolerance reasons. Maybe the patient's not well enough, they have heart conditions, things like that, that they can't have a major operation. So, in those cases where it's a localized problem, you know, it's obviously better to give a localized solution for that. And so we have things like radiation, but those too are limited. There's certain tumor types that don't respond to those. Additionally, there could be other radiosensitive organs and things around it that just won't tolerate that. So having a local option where we can deliver, an immuno-oncology approach localized to the tumor, I think is incredibly attractive. So the other is, by injecting it directly into the tumor, you really concentrate that drug therapy there. Um, that also, I think that initial experience with SNV1 will help us to, or Supernova will help us to, build faith that this drug really is optimized in a way that is effective. But following on that with RT Nova, there are other patients who have lesions that are not reachable by a needle, at least not safely. And so having an option that can be delivered systemically, I think really will open up a virotherapy to a broader range of patients. And again, the ability to adapt it from a payload standpoint adds another level of versatility to the drug. So to me, these therapies both complement each other very well. And there will be a patient, an optimal patient for each of these therapies at the end of the day.
What opportunities do you see for strategic partnerships and collaborations that could accelerate Calidi's growth and enhance shareholder value?
Sure. So for both Supernova and Neuronova, I think that there are real opportunities for combinations with other approved or even novel therapies. And so some of this comes back to our biomarker strategy, as part of this trial is really around getting those biomarkers, understanding, what other therapies could be used in combination with these drugs to really enhance efficacy further. Now, it's unclear that we'll need that. I think, these drugs in and of themselves are effective, but there's always a possibility for partnership to gain even greater efficacy in combination for that. For Neuronova, I really think there are, or sorry, for Artinova, I think there really is quite a few opportunities for partnership, and that's something that we will look into. The ability to have an adaptable payload, we have thoughts and we're advancing our own ideas as far as what we think the right payload is. But obviously, payloads that come from, um, other pharmas, for example, the ability to take a therapy that maybe did not advance because of systemic toxicities if you deliver it at high doses through the blood and have it really delivered in the tumor microenvironment by a viral payload, to me, I think really opens up drugs that we know are effective but just aren't well tolerated. And I think that's going to be very attractive to other biotechs, large pharma, to partner with. So I think that's certainly an opportunity we intend to capitalize on.
And finally, what metrics or leading indicators do you believe best reflect Calidi's progress, particularly in light of platform validation milestones such as IND acceptance or biomarker-driven clinical response?
Yeah, I think IND approval is certainly a big milestone. Um, we were able to get this. Really, we had no concerns from the FDA, from our all the aspects of the drug, both clinical trial design, CMC. So from that standpoint, that I think that's a big milestone. And then the other is really the company is assembling the right expertise, not just to advance this in the clinic, but with an eye towards um, the next steps beyond this phase 1 trial. So to me, these are all really key leading indicators that we're moving to a clinical stage company in a way that's going to ultimately have us positioned to generate value, as quickly as possible. These things do take time, but I think we've got all the right, pieces in place to advance this as quickly as possible. And then along those lines with RT Nova, I think this, we're advancing that preclinical asset really very quickly because we think it's so important and novel that we want to make sure that we've done all the necessary work ahead of time, but at the same time, we want to move it into clinic as efficiently and thoughtfully as possible.
And now 3 questions for Andrew. What is Calidi's current financial position, and how does leadership plan to secure the funding necessary to drive clinical trials and commercialization efforts?
Sure. So we finished December last year with 9.6 million cash in the bank. Then we did 2 public offerings in the first quarter where we raised over 8 million in gross proceeds. So we have investors that really like our story, that really see the value in what we're doing. And then in addition to that, we got $3 million from CIRM, that's the California Institute for Regenerative Medicine. So that's non-dilutive funding in 2024. And that was for preclinical work. And now with an open IND, we can apply for another 8 million in funding from CIRM for our clinical work on CLD 201.
Can you tell us whether you've increased or reduced your debt?
Yes, we've actually drastically reduced it. So in 2024 and the first week of 2025, we converted to equity or paid off over 8 million in debt. So after that first week in January, our consolidated balance sheet would show about under 2 million in debt. So a big reduction.
Finally, Andrew, for investors looking at the biotech space, how does Calidi's unique approach to cancer therapy translate into opportunities for partnerships or licensing?
Yeah, that's a good question. And I think Travis hit on this as well. It's the ability of RT Nova to deliver that transient payload to targeted tumors. So with that, we can deliver any therapeutic protein, a checkpoint inhibitor, a cytokine, to the targeted tumor. So that's what really opens it up for strategic partnerships or collaborations.
And Andrew, if you would like, you can now begin reading the questions that have been submitted by our audience.
This one would be for Travis. I'm not sure if we're in a position to answer this or not, but can you give us a range of possible payload for the first drug candidate?
Yeah, I think I can speak to this broadly. We have lots of ideas on this topic, and some things we will um, publish or put out in the near future. But for now, things that are going to modulate the tumor microenvironment to augment an immune response sort of makes inherent sense. But it isn't limited to that. If you think of both toxins and chemotherapeutic things, like all these things are possible. And so really, the range of possibilities is so broad that it's both exciting, but we're going to focus on the areas that we think, are most impactful initially. Some things, we do have work to do to understand how specific, and we've done a lot of those important works, but that's going to impact the payload. We've done a lot of this preclinical work already, and so let me just leave it at that. I'll keep talking because I think the short answer is it's a really broad range of things, and so but we will put out some specific information to that in the near future.
Great, thank you. I'm going through these questions, and I think we've answered most of these questions that were related to either financing or the science. So I don't think we have any further questions.
All right, and thank you very much, Calidi team. Just before we sign off, for more information about Calidi Biotherapeutics, you can reach us at 1-800-REDCHIP or email us at cldi@redchip.com. Please visit RedChip's investor information page for Calidi Biotherapeutics. It's cldiinfo.com. There you can view and download the investor presentation and fact sheet and sign up for news alerts on Calidi. Thank you again to our many participants today, and thank you very much, Calidi team.
Thank you to everyone for your interest.